Hello everybody and welcome back to the Biomolecules. So last lecture we have talked about the structure of the proteins, how a protein assumes its secondary and the tertiary structure. So once again a quick recap that you have a primary structure of the protein that is governed by the sequence of the amino acids, and we have seen that if you draw a amino acid sequence in this fashion, then there are two different kinds of angles that can be forming. One is for the single bonds. So one is this bond which is termed as phi, other one is this bond which is termed as psi. So these two bonds are of course in different plane, and there is an angle that is created between the planes. This is called the dihedral angle. And so if you rotate one of them fixing the other, then you change the torsional angle, and therefore the structure of the protein gets changed. So this is the primary structure of the protein, which essentially means the sequence of it, the amino acid, specific amino acid sequence. Now the secondary structure of the protein, which we have seen that it can either assume alpha helix or beta sheet, that comes from the rotation around the phi bond or the psi bond. So as we have seen here, and that you can calculate by using the Ramachandran plot. Ramachandran plot essentially tells you that which rotations of the phi angle and the psi angle are actually allowed, and there is, you can put dots or the points for all the rotations. If you make a plot of this, is the phi goes from minus 180 to plus 180, which makes it 360-degree rotation. Similarly, the phi and the psi goes from minus 180 to plus 180, that makes it again a 360-degree rotation. So if you try to rotate them simultaneously or keeping one fixed and rotating the other, then you will have a plot like this, where these colored points basically tells you that which rotations are energetically allowed. And those will of course, if your density is high, then that means those kinds of rotations will give you a stable structure of the protein, and that is what we call the secondary structure of the protein, which is the alpha, either mostly the alpha helix or the beta sheet. There can be different kinds of alpha helix, there can be different kind of beta sheet formation. So if you assume an alpha helix structure, it will look like maybe something like this. So where if you rotate one, then it will come to such a position that the peptide bond, one carbonyl group of the peptide bond and the amine of the other peptide bond may come in close proximity and form the hydrogen bond. Similarly, it can be here NH, this is C from the backbone, and they can come in hydrogen bonding formations. That makes it a alpha helix. Similarly, here can be a carbonyl, here can be NH, and these are the NH and the CO of the backbone, the peptide, this NH, this CO. Now this forms the alpha helix structure. If I consider one of this bond, so R if you consider, and then what you have is here NH and a CO, NH, and it goes like this, and then you have a CO goes like this. So if I make, here is your R, here is your N, and this H, C, here is your CO. It assumes this kind of orientation, let us say. So then if you look at the distance between the NH and the CO, so this is around 0-degree angle. The distance between the NH and the CO would be like this here, and they can come close in proximity and can form maybe hydrogen bonding, or may not be. I mean, they may need to come more closer sometimes. Now technically, if I just make a 180-degree rotation, then this CO, I am rotating around this bond, gets flipped off, you still have the R and you still have the NH like this. Now, you see the distance between the CO and the NH gets completely opposite, and that creates the change of the secondary structure. So, here there cannot be a hydrogen bonding between this NH and this CO, but it can assume a hydrogen bonding with another CO of a second strand. So, if you consider another molecule of the same protein, it can have a hydrogen bonding with that. Similarly, if you have the other strand, they can form hydrogen bonding. So, that is the difference between the changing of angle. By changing the angle, you change the geometry of the primary structure, that brings you to the secondary structure. So, typically if your phi and psi angle, as I think I have mentioned it in the last class, is around minus 60 degree, then you have a alpha helix formation, because it will rotate it and bring it to that level where the NH and the CO can come close in proximity. And of course, this is governed by the sequence, what sequence the peptide has or the protein has. And very commonly, if you have alanine, leucine, methionine, phenylalanine, and there are more, they typically tend to form alpha helix kind of structure. And if your phi versus psi angle is around minus 135 degree, then that assumes a zigzag structure of the protein, and that means they are prone to form beta sheet structure or beta sheet, as I will call it, assembly, because it happens between two molecules or more than two molecules. So, this is basically a supramolecular architecture or supramolecular arrangement of the different protein molecules. On the other hand, alpha helix or the hydrogen bonding that is that exists within the alpha helix is more intramolecular process, and beta sheets is more intermolecular hydrogen bondings, and that makes it a supramolecular self assembly. So, relatively larger amino acid like tyrosine, tryptophan, and the crowded ones isoleucine, hydrophobic, crowded, hydrophobic with branched chains, isoleucine, valine, cysteine. These kinds of amino acids, if they are present in a sequence or if the protein is rich in such sequences, then those segments will be more prone to form beta sheet kind of structure. So, within a protein, if you consider a protein, within a protein, a certain segment of the protein can assume alpha helix conformation, certain other part of the protein can assume beta sheet conformations also. Not necessarily the whole protein will be either alpha helix or beta sheet, no. So, that is the secondary structure of the protein, alpha helix or the beta sheet, and this is formed, as I have mentioned, due to the short range interaction. Primarily hydrogen bonds. Now, once a secondary structure is formed, it undergoes further modifications to assume a tertiary structure of the protein, which is basically the functional protein. So, the tertiary structure means it is the three-dimensional structures or the folded structure of the protein, which comes from coil formation. So, the protein will further mold into a folded form or a coiled form, that is known as the tertiary structure, and this is because of relatively long range bonding interactions. That assumes the tertiary structures. I will just show you in a brief period how the interactions will take place, what are the long range interactions that happens. This is mainly, for example, that if you have secondary structure which is the alpha helix here, and then if you have a polar group or let us say an amine functional group here, and then a carboxylate functional group side chain in this case, and then they want to come in close in proximity because of the electrostatic interactions. Because the amine group is positively charged, acid group is negatively charged, so they want to come closer together. So that will force this helix to go further fold, so that the negative charge and the positive charge can come closer and form a electrostatic bond. Similarly, there are other types of interactions that I will just briefly explain very quick. So, these are the long range bonding interactions that forms a tertiary structure of the protein. Now, apart from the tertiary structure, there is another structure that some proteins have, that is known as the quaternary structure. Quaternary structure is basically, if a protein, sometimes a protein has many subunits, more than one unit of a protein. There are other units, we call them subunits, or they can be called oligomeric proteins. So, those different subunits of the protein are interconnected, of course. So, the way they are interconnected and form a total structure is known as the quaternary structure. So, it is basically, the quaternary structure is basically the assembly of the subunits of a protein. So, as you know that when a protein is formed, protein is usually biosynthesized in our cellular systems, in the living cells. How is it biosynthesized? All the details, we will have a separate chapter for it, that it comes from our genetic code, that is DNA, and then it goes to formation of a messenger RNA, or known as mRNA, and from mRNA it is transferred to a tRNA, and then the tRNA synthesizes the protein. So, when there is the synthesis of the protein, of course, it synthesizes the covalently linked primary structures, the sequence of the protein. So, once a sequence of the protein is synthesized, then depending upon its sequence, it will automatically resume or assume the secondary structure, and then the secondary structure will go to the tertiary structures, that is the functional protein. So, once a tertiary structure of a protein is formed, then only it can do a biological transformation, or it will have a certain biological activity. And of course, again and again I am repeating that what kind of tertiary structure a protein will assume is always dictated by the primary structure of the protein, that is what sequence it has. Now, there are a lot of studies that has been done or still being carried out to understand how the protein is folded into that kind of specific structure. Why not anything else? Why not a slightly different type of three-dimensional structure possible for the protein? Similarly, how you can unfold a protein from the tertiary structure to its primary structure, and to study the dynamics of all these processes. So, we call it typically a denaturation. When a protein is unfolded, that means you are breaking all the interactions that is present in the tertiary structure and then bringing it back to the kind of primary structure. So, that is the process is known as the denaturation of the protein, or you can also call it unfolding of a protein. So, the moment you unfold a protein or you force a protein to denature, its three-dimensional tertiary structure is disrupted, and therefore, the protein cannot function anymore. This is called the inactive protein, and we will see just in a brief time that folding of a protein from the primary sequence to the tertiary structure is a spontaneous process under physiological condition, and of course, the primary structure determines the tertiary structure of the protein. So, now what kind of interactions are present within the tertiary structure of a protein? If you understand that, then only you can think of breaking those interactions and make and unfold or denature a protein. You can find a suitable way how to denature a protein or how to unfold a protein, and then can find again methods to study the dynamics of those processes. So, if I take an example of a tertiary structures, I am just drawing anything arbitrary like this. So what are the forces that makes a tertiary structure, or what are the bonding interactions that forces a protein to assume a tertiary structure? One of them is of course the interactions of the side chains. Side chains can be of very different types. As we have seen, we have 20 amino acids with very different functional groups, very different properties. So depending upon the side chains, the interactions can vary also. So one is, if you have the electrostatic interactions, electrostatic interactions. What does that mean? So when you have seen that secondary structure, you have seen the hydrogen bondings were present within the strand for alpha helix. For beta sheet, it was between the molecules. So here further, for the side chain, if I consider, for example, I consider a lysine. Lysine has amine side chain. There are 4 CH2 groups. I am not drawing them, and there is amine here, and this amine assumes, as you have seen, into its positive charge form. Now if you have closer in the opposite direction somewhere, if you make it a flat structure, you will see the distance between this lysine and this residue here is actually very large. You have to go all the way here, but because of such interactions, like if you have aspartic acid, for example, aspartic acid is CH2 and then you have a CO, then you have OH, and this OH of course exists in O minus. So now you have a positive charge, you have a negative charge. So if in the secondary structure, if there is a chance that this lysine and the aspartic acid somewhere else are to some extent in close proximity, then in order to form a better bond, in order to form electrostatic interactions between the positive charge and the negative charge, it will force the strand to mold more and bring the positive charge and negative charge within the bonding interactions. So that forms the tertiary structure. And of course this is a long range interactions, as I am saying, because if you look, the distance between this and this is quite long, but the spatial distribution or the spatial arrangement forces these two residues to come closer together, and that folds the protein. So one reason for assuming the tertiary structure is electrostatic interaction. Second, if there is electrostatic interactions, and then there will be the opposite one, the hydrophobic interactions also. Hydrophobic interactions means, so when a protein is in a solution, typically within the biological cells, as I have said, that it has a pH of 7.4, so more or less, and it is an aqueous medium. So protein is in an aqueous medium in a pH of 7.4 roughly. So there are water molecules surrounding it, and hydrophobic side chains of the amino acids, they repel water, so they won't want to be exposed towards water. Their tendency will be to go away from water and hide. So typically that happens here. So if you have multiple number of hydrophobic residues or hydrophobic side chains, for example the valine, isoleucine, and maybe phenylalanine and so on, so they would want to go or hide away from water, and they will form a core. So if you have for example a valine here, I am just making another coil, so valine here, CH3, and maybe another valine very close here. If you have phenylalanine here, so they will all try to gather themselves to form a hydrophobic core. It is called the van der Waals interactions or hydrophobic interactions. So, water molecules are outside, all outside here, all water molecules surrounding the protein. So, they will shy away from the protein, from the water, and want to go inside the interior of the protein. And once they go inside the protein, they have the hydrophobic molecules also have the hydrophobic interactions within them. So, that is a stability factor. So, second is hydrophobic interactions. A third one, very common actually, is formation of the disulfide bond. So, if you have a cysteine, so this is cysteine within one protein molecule, and let us say there is another cysteine maybe within the same protein, or can be in a different protein, molecule. So, CH2 and SH. So, these two, if you take a cysteine and keep it in air, so it will immediately undergo oxidation. So, redox reaction basically that will form CH2S, CH2, and here is the this CH2, and here is the rest of it, CO and the NH and the rest of it. So, it will form a disulfide bond, and that is a covalent bond. So, quite strong bond. So, cysteine residues, if they are in close proximity, that will tend to form a disulfide bond if you keep it in air under aerial condition or an aerobic condition. So, and that will further force the helix or the beta sheet to mold it and bring them closer so that they can form the covalent bond. So, here if you have a SH residue or the cysteine residue on the same strand, somewhere else if you have another cysteine residue, and if they are in open chain, so they will fold themselves and form the disulfide bond. That is the driving force. The formation of the disulfide bond is the driving force for molding the structure. And there are other interactions. In the next slide, I will show you with proper examples. So, these are primarily three types of interactions that dictates the formation of the tertiary structure. So, I will give you an example how the disulfide bond is formed in protein. So, a very well studied protein actually is bovine insulin. You know, insulin is a medicine that is used to treat the diabetic patients, right? It is basically an enzyme. So, bovine insulin was actually the insulin which used to be used as the medicine to treat such diabetic patients. Nowadays, we can manufacture insulin from human sources itself after the discovery of the recombinant DNA techniques. So, bovine insulin is a very well studied protein, and that shows multiple number of disulfide bonds. So, it has two chains, it is called A chain, and that has a sequence. I will just write, I am writing the part of the sequence: glutamic acid, then glutamine, then you have a cysteine, another cysteine, alanine, serine, valine, then a cysteine, then a serine, and it goes, and there is another cysteine at the position 20. So, this cysteine position is 6, This is 7th position and this is 20th position of the cysteine. Now, this cysteine has a SH residue, this cysteine has a SH residue, and they can form the disulfide bond that locks the protein structure. Similarly, the second cysteine, cysteine, alanine, serine, valine cysteine. Here there is SH, and this S and this S can form the disulfide bond, and therefore the protein structure has to mold or has to coil so that these two can come within the bonding interactions. Similarly, this has a SH, and in the next chain of the protein molecule of insulin known as the B chain, there is another cysteine which comes closer to it, opposite. So, before it has a leucine, and then histidine, and then a glutamine, and so on. So, this is also the 7th position of it. And then here there is another cysteine, and this position is 19. So, this cysteine has thiol, this cysteine has a thiol, and they form disulfide bond very quickly. Similarly, the S of this, they form disulfide bond very quickly. So, that overall structure has many disulfide bonds, and that is the driving force for the tertiary structure of the bovine insulin. So, here I have made a schematic presentations of how different interactions are present that makes a tertiary structure of the protein. So, this is, for example, this is the secondary structure of the protein. Secondary structure of the protein, and it gets down to the tertiary structure of the protein, which is the coiled form or the folded form. So, basic laws of chemistry that drives the protein folding are: first is the hydrophobic side chains that has been given the color as black. For example, this and this, they are the hydrophobic side chains, as I have mentioned, valine, isoleucine, leucine, phenylalanine, all those. They do not have functional groups or polar side chains. So, they are termed as black here. They will be buried on the inside of the globular protein, where they are hidden from polar water molecules. Water is polar and they are surrounding the protein molecules, as I have explained. So, these things, when they form the tertiary structure, they want to hide away from water and forms this core kind of structures. So, that is the driving force for the protein to fold in this fashion. Second is the charged side chains, means it can be of two charge, one is positive charge, other is the negative charge. Negative charge is for the acid residues like the aspartic acid and glutamic acid. The positive charges coming from the amine residues, for example, the lysine and arginine, both have amine side chains. So, blue and the red are marked for positive and negative side chains. So, they will be on the surface of the proteins, because since they are charged species, they would want to interact with water molecules also, because water can be is polar, and of course, the charged species are also polar. So, they primarily would be exposed towards water, and of course, they will have the interactions within them, bonding interactions or electrostatic interactions within them, positive and negative. Polar side chains other than the charged ones, which has polar functional groups, will be again on the surface of the protein. They would be on the surface. So, that is represented by green dots. They would be on the surface of the protein, because that is the part where they are exposed to water molecule. So, they can interact with water molecule and get solvated. That is a very effective stability factor, the solvation energy, when a molecule is solvated or surrounded by water molecules, and that is a highly energetically favorable process. So, the polar side chains will be on the surface of the protein, and they can form the hydrogen bond with the water, that is the solvation factor. Fourth is the cysteine side chains. Cysteine is I have given it as purple color here for a bond formation, SS bond formation, that often interact with each other to form covalent disulfide bonds that stabilize the protein structure. So, these are the factors that are responsible for the formation of the tertiary structure of a protein. And obviously, now you can see how the primary sequence or how the primary structure of the protein, the sequence of the amino acids are dictating the folding of the protein depending upon their side chains. So, a classic study of how the protein folds and unfold was done by Professor Anfinsen. This is called Anfinsen's experiment or Anfinsen's protein denaturation and folding. So, Anfinsen received Nobel Prize in 1972 in chemistry. He has been working for a very long time on the enzyme ribonuclease, RNase. He was one of the key persons who has worked a lot on RNase, the ribonucleases, and a part of his research was of course to see how the tertiary structure is formed in the protein, why it is such, why does it assume such specific structure, how you can unfold a protein and can inactivate a protein. So, he has done a complete study and dynamic study for the protein denaturation and refolding. So, it is a sequence of process that first studies. In order to study the folding of the process, what he has done, he has first tried to denature the protein. And how can you denature the protein and then refold the protein again? So, we have seen what are the forces that are involved in the tertiary structure. Now, if you want to denature a protein, then you have to break all those interactions, and therefore, now you can find a way or think of a way how can you denature or how can you break such kind of interactions, then only you can denature the protein. One of them is of course heat. You can heat the protein high up and then break all the non-covalent interactions, but it still has the problem that sometimes the protein degrades by itself, or there are other bonds, as you will see, that especially the disulfide bonds and others, which are hard to cleave by heat. But there are other reagents which you can cleverly use to cleave those bonds. So, denaturation of a protein means the loss of tertiary structure. Once the tertiary structure is lost, it will lose its activity. So, what Anfinsen found out that if you use urea, then you can denature a protein. Urea is an agent, very simple reagent of course, is a reagent that denatures the protein. It disrupts all the non-covalent bonding interactions present within the protein, and therefore, you break the native tertiary structure as well as the quaternary structure. But after denaturation with the urea, you still have the disulfide bonds left which you have to cleave, and therefore, the second reagent is coming, beta markup to ethanol. So, very simple reagent, structure is very simple. What it does is that it is a anti-redox, I mean it changes, it undo the process of the disulfide bond formation. So, when you have the cysteine here and a cysteine there in the protein, they formed a disulfide bond. Now, if you treat these with beta markup to ethanol, this will reverse the process. It will make plus 2 individual cysteine by cleaving the disulfide bond, and in the process this molecule itself so this is also this has also a thiol bond. Two of these molecules will undergo disulfide bond formation, OH, S, CH2, CH2, OH. So, this will form the disulfide bond and undo this process. So, it is a redox reaction as I have said. So, this will be oxidized and the protein will be reduced to individual cysteines. Therefore, you can break the cysteine-cysteine disulfide bonds also. Here is how the process goes on. It is done in two steps. The first step is treatment of the protein, that is in this case RNase, with 8 molar of urea. And Finson has found out that the concentration of urea is optimum if you use it on 6 to 8 molar. Urea is ideal for denaturation of the protein. So, that is the optimum condition to denature the protein. So, once you use urea 6 to 8 molar, then it breaks all the non-covalent bonding interactions such as the hydrogen bondings that were present. Hydrogen bondings were present within the secondary structure also, as I mentioned, as well as in the tertiary structures with the water molecule within them, within the molecule, as well as the polar, if you have the polar side chains and they form hydrogen bonding with water also. So, lots of hydrogen bondings will be present within for the tertiary structure of the protein itself as well as the secondary structure of the protein. So, urea will break all the hydrogen bonding interactions. At the same time, it will break all the charged interactions, all the other van der Waals interactions and the electrostatic interactions also, like the charged species lysine with the aspartic acid. Those kinds of interactions will all be broken by using urea. But still, once you destroy all the short range interactions or the long range bonding interactions, you still have retained the disulfide bonds. So, therefore, after treating with urea, it partially denatures the native tertiary structure. Second, as I said, is a treatment with the beta mercaptoethanol. It is a redox reducing reagent that will oxidize itself and reduce the disulfide bond back to the cysteines. So, once you treat this with the beta mercaptoethanol, then you have completely disrupted the tertiary structure of the protein and that will lose the native structure. Therefore, complete denaturation will happen. And once you denature the protein, then the protein, or in this case the RNase, is completely inactivated. It cannot do its biological function. That is what Finson has found out and have studied. So, after this treatment of these two consecutive process, Finson has found out that RNase, the enzyme RNase, does not retain its biological activity. The question is, can you refold the protein? Can you make the protein functional again? So, that is what is very interesting study actually. Once you denature a protein, can it assume its activity, biological function again, which means the unfolding structure or unfolded structure and the folded structure of the protein, do they exist in equilibrium? So, that is what he has also studied. Now, in order to refold the protein, what you have to do? So, you have urea in it. By using urea, you have disrupted all the short range interactions or all the weak interactions, weak bonding interactions, non-covalent interactions I mean. So, unless and until the urea is present, you cannot regrow those bonding interactions. Similarly, you have used beta mercaptoethanol to break the disulfide bond. So, you have to remake the disulfide bond again and remove the beta mercaptoethanol. So, two things you have to do. One is you have to remove the urea from the solution and you have to also remove the beta mercaptoethanol from the solution and then make the disulfide bond again. So, that is what he has done. So, slow removal of urea, that is the first step for the refolding process to bring back the protein. Slow removal of urea. If you I do not know whether you are aware of this process called dialysis. Dialysis is a process where you have a membrane. If you have a vessel full of water or medium, Then you take a bag. The bag is made of it's a membrane basically, semi-permeable membrane, semi-permeable membrane. That is what we use as dialysis bag. So, semi-permeable membrane if you have, then what it will do, and if you have some content inside the bag in a liquid form, then what it will do, it will allow only water molecule or the very small molecules to get into it or to come out of it. So, a semi-permeable membrane allows the permeation of the small molecules. That depends on the molecular weight of it. Usually the low molecular weight molecules can get into the membrane or can come out of the membrane. So, protein has a very large structure. They have their molecular weight in terms of kilodalton, and on the other hand the urea, beta mercaptoethanol and all those reagents that you have used, they have very small low molecular weight small molecules. So, if you use a semi-permeable membrane, that will allow the larger molecules like protein to stay, not to flow out, go in or out, but allow the small molecules to come in and out. Then what will happen? Water molecules will get into the membrane, water molecules can get into the membrane, and depending upon your chemical potential, that will allow the smaller molecules like urea to come out of it, because the concentration of, it's a concentration gradient, right? Water is pure water, there is no urea in it, and inside it you have a high concentration of urea. So, in order to balance the concentration of urea, urea will come out over time. So, if you do a dialysis over time, then the smaller molecules because of their concentration gradient will come out of the dialysis bag where your sample was kept. So, if you do a dialysis through this process, you can remove the urea, the excess of urea that was present. Similarly, you can remove the excess of beta mercaptoethanol that was present in your protein solution. So, now you are, after the dialysis, you are devoid of urea, your protein content does not have urea anymore. So, once urea is gone, now the bonding interactions, the electrostatic interactions, the polar interactions, the hydrogen bondings can again take place. And after the removal of beta mercaptoethanol, it cannot do the reduction again. If you have the cysteine and cysteine that was reduced by using the beta mercaptoethanol, it was reduced to pure cysteine. So, once you remove the beta mercaptoethanol, it cannot do this process again. Whatever has happened. Furthermore, it cannot do the reduction. So, whatever cysteine, free cysteine that was present and the other cysteine that was present will remain. Further reduction will not happen. So, once you remove the beta mercaptoethanol and if you allow it to oxidize again, this is done by aerial oxidation. So, after the dialysis, if you take your protein content and allow it for aerial oxidation, then this process will again resume. Cysteine SH will again form the disulfide bond. So, you are allowing the protein again to form the disulfide bond, the covalent bond, and therefore, it can regrow its tertiary structure again. So, oxygen oxidizes the SH groups to disulfides, refolded in absence of the reducing agent, that is the beta mercaptoethanol. And once you do both the treatment, then protein is again refolded and regained its activity. That has been studied, that after you remove all these reagents that you have used at the beginning, the protein resumes its active biological functions and activity. So, that is the study done on, there are a lot of studies, of course, this is one of the famous studies that has been done to understand the protein folding and unfolding kinetics or the dynamics, or whether it is a equilibrium process or not. And here comes the thermodynamics of the process. So, you have the secondary structure of the protein coming to the tertiary structure of the protein. Secondary structure is the inactivated form, secondary structure of the protein coming to the tertiary structure of the protein. Secondary structure is the inactivated form or the unfolded form, and then when it is the tertiary structure, this is called the native form or the folded protein. Tertiary coiled structure, secondary structure has a random structure. And if they stay in equilibrium, then there is equilibrium constant, which is defined as K in this case. Now, the question is whether the process will move forward direction or whether it will go backward. In other words, is the folding of a protein is thermodynamically favorable process or not? So, that you can actually understand or that you can explain from studying the delta G of the system. So, as you know, whoever has the basic thermodynamic equation is delta G equals to delta H minus T delta S. So, delta G is the Gibbs free energy for the process, delta H is a change of enthalpy, and delta S is the change of entropy. T is the temperature at which you are doing the experiments. In this case, it is the room temperature or the biological cell temperature, which is 37 degrees Celsius. Now, as you know, in order to make a process in the forward orientation, if you want to go in this forward direction, then the delta G of the process of the overall reaction has to be negative. So, Gibbs free energy has to be negative if you want a forward direction inactivate to protein to activated structure. For any spontaneous reaction, delta G has to be negative, that is called the Gibbs free energy. Now, let us see whether this process has a negative delta G or not. So, in order to calculate delta G, you have to think of delta H and delta S. Separately, if you see the change of enthalpy delta H, what do you gain or what do you lose by this process, unfolded to folded process? So, in the folded tertiary structure, you have polar interactions, as I have mentioned, and that is a favorable interaction. So, delta H would be negative there. Hydrogen bonding again makes a stable structure, releases energy, that will give you negative delta H. Hydrophobic interactions again will stability factor, that will give you negative delta H. Solvation energy is a huge stability factor, and that will release lot of energy. So, delta H would be negative for this process. And then steric repulsion is a instability or distability factor, that will require some delta H. So, overall the delta H for the folding process is favorable, or you will have negative value of delta H in this case. So, now think of the delta S, change of entropy. So, entropy means the degrees of freedom, whether the molecule are flexible, whether the molecules have the freedom to move around the solution, whether the atoms within the molecule have higher degrees of freedom or they are free to move. So, in a locked tertiary structure, obviously the degrees of freedom is restricted because of different kinds of bonds and the bonding interactions, as compared to the flexible helical structure or beta sheet conformations or even the primary structures can assume to random conformation. So, change of entropy is a loss of degrees of freedom for the folded structure. So, it is an unfavorable conformation. And locked conformation, as I have said, that the conformation, because of disulfide bond here, the conformation is restricted, the protein cannot move. So, entropically it is not favorable process, but enthalpy wise it is a favorable process. Now, delta G equals to minus RT ln K if you want to calculate the equilibrium constant K of this process. So, K is the equilibrium constant, K equals to e to the power minus delta G by RT. R is fixed, T is fixed, because we are doing the reaction at room temperature, at a fixed temperature. Variable is only the minus delta G. and when you calculate minus delta G in terms of delta H and delta S, delta H is favorable, delta S is not favorable. And the experiment says that overall you get around change of delta G of this process, unfolded to folded process, is about 5 to 10 kilocalorie per mole, a stability factor. So, delta G is minus around, I am taking just 10 value, 10 kilocalorie per mole for the protein folding process, which is actually a very small quantity, very small amount of change of energy between the unfolded to the folded structure, only 10 kilocalorie per mole, which means if you have, this is the folded form, and if you have, this is the unfolded form, then this difference of energy, if this is your energy, is only roughly 10 kilocalorie per mole, very small change of energy. Now, if it is minus 10, then it comes down to e to the power minus 10 kilocalorie, means 10 into 10 to the power 3 calorie divided by R. R in terms of calorie is 2 calorie. So, 2 calorie will cancel each other, into temperature, room temperature is 298 degree, 8 Kelvin. So, this value will come down to around 2 into 10 to the power 7, that is K. So, this is K equals 2 into 10 to the power 7. So, you can see a very small change of energy here brings a large change of equilibrium constant. 10 to the power 7 is a high rate of conversion. So, this process from the inactivated protein to the folded structure of the protein has a huge equilibrium constant, which means it will move forward this direction spontaneously. So, in other words, if you calculate that from here 2 into 10 to the power 7 equilibrium constant means if you have one molecule of the unfolded form, then you will have around 2 into 10 to the power 7 number of molecule in the folded conformation, which means the most of the protein molecules, if you keep it in the solution, it will stay as folded conformation, folded structure or the tertiary structure. In other words, the formation of a tertiary conformation or tertiary structure of a protein is hugely thermodynamically favorable process. So, now I will move on, protein is I am completing the protein part. Now, I will quickly move on to the fatty acid structure and lipids and then complete the module. So, fatty acids I have described in my first lecture or the second lecture. The fatty acids have this kind of structure where you have a carboxylic acid terminal, that is polar, and then a large hydrophobic side chains, and this side chain can vary in number of carbons. In terms of number of carbons, here you can change the number of carbons are changing, number 1. Number 2, they can have double bonds. They can be saturated fatty acids or unsaturated fatty acids. So, these are some example of saturated fatty acids. For example, this is palmitic acid, stearic acid, these names maybe you have heard. And then there are unsaturated fatty acids. Most natural unsaturated fatty acids are usually have cis conformation. So, this you see, this is a cis conformation. This is a trans conformations. This is cis, this is trans. Cis and trans conformation. Trans conformation is actually bad for health. Trans fatty acids are bad for health because they are not easily digested. Cis fatty acids are easy to digest because they are easy to convert into doing chemical reactions is easy for the cis forms. So, cis fatty acid this is oleic acid, and this is with two cis unsaturations at 9 and 12 positions is linoleic acids. These are also known as omega fatty acids. If you know, omega fatty acids are good for our health. So, these are some of the fatty acids which has the simple structures with the carboxylic acid terminal, and then you have the triglycerides which have branched chains, 3 hydrocarbon chains and a glycerol moiety here, and then you can have the mixed triglyceride with different carbon chain lengths. There can be unsaturations also here. So, these pictures I have taken from the book Miller and Tanner. So is this, and there is other example of phospholipids. Phospholipids have a phosphate group here and a lipid moiety. Lipid moiety means basically triglycerides that has side chains. In this case, one of them is a phosphate. So the hydrophobic side chains contains unsaturations. This is one example, and then this is other example of the triglycerides. This is called glycolipids. Glycolipids has a glycosidic bond with a sugar attached to its carbohydrate moieties. It can have more chain lengths of carbohydrates also. It can have single unit as well. Of course, it has a phosphate, but not necessarily, depend upon the molecule, and then it has 2 hydrocarbon chains. Another is of course the cholesterol. Cholesterol has a ring structure and all hydrophobic. So, most of these lipids, fatty acids are present in our fat. Of course, you know, now lipids, different kinds of lipids that I have shown, they form the membrane of the cells, and that is how our cells are very well protected, because they are mostly hydrophobic in nature. So, they make a thick layer, and they do not allow the foreign bodies to enter into the cells. I will just here you can see how the layer is formed and how they repel the molecules to enter into the to cross the cell walls. That is how we are very well protected. So, lipids, they are basically the biomolecules, and they are insoluble in water, insoluble because of course they have the long hydrocarbon chains, but they are highly soluble in organic solvents. You can make them soluble in various organic solvents, and they are primarily they constitute the membrane of cells. So, major membrane lipids, those are phospholipids that I have shown. It contains a fatty acid as well as and a phosphate moiety. It contains a glycolipid. Glycolipids are the lipids which are covalently conjugated to carbohydrates. One or more chains of sugars can be present. This is the sugar chain. One or more sugar chains can be present. That is the glycolipids. It is a major constituent of cell membranes. Then the third one is the cholesterol. Cholesterol is a steroid or fat primarily produced in the liver or intestine. It is present in intestine and liver also. And the membrane lipids, they usually form a bilayer sheet kind of planar kind of structure in water compared to micelles. So, if you know how a micelle is formed, if you have a fatty acid for example, and you have the OH or O minus. So, this is the polar head, and this is the non-polar part, and we call it a tail. So, if you put the fatty acid in water, then what will happen? The polar head part would want to interact with water because it is polar, water is also polar, but the tail part, the long hydrocarbon chain, would want to go away from water because it is a hydrocarbon, and therefore it will repel water, will not be soluble in water. So, as a result, it will form a aggregation kind of thing, self assembly or aggregations, where you can see such kind of structure will be forming. Here all the water is there, and therefore, the polar head would be exposed towards water. On the other hand, the hydrocarbons would shy away from water, and they would want to move away from each other and make a core here. This is how a micelle is formed, and this is a characteristic of most of the fatty acids. They form micelle in water in appropriate concentration. So, on the other hand, in membrane, instead of micelle, they form a bilayer kind of thing, because if you look at the structures, they are highly branched structures. So, micelles will be very hugely crowded and there will be steric crowding there. That is why they primarily form the bilayer. This is how a bilayer would look like. The polar head here, this is the hydrocarbon chain. For schematic presentation, two have been used, hydrocarbon chain. Naturally, the other molecule, the hydrocarbon will interact with the hydrocarbon. So, the opposite direction another layer would be formed, and since this is polar, of course, this is exposed to water here, this is exposed to water here. So, bilayer in the sense that you have water, then you have a lipid layer which is non-water, and then a water. That is why it is called a bilayer. That is how the membrane is formed. So, a cellular membrane basically is this, which is hydrophobic layer, outside water, outside water. Now, a molecule cannot, most of the molecules that are soluble in water, they will not allow to cross the cell membranes, and that is how our cells are protected. So, now I am towards the end of the module 1. So, I will give you a rough synopsis of whatever we have covered in this module. So, we have seen at the beginning that the discovery of DNA structure and how the function of DNA was elucidated from the structure of it. So, elucidation of structural parameters of DNA and RNA both. So, we have seen how the different conformations, different orientations, different angles, and the functional groups like sugars, nucleobases, they are playing role in constituting the final structure of the DNA, the tertiary structure of the DNA, that is the bDNA structure. How double helical DNA explains cell division and carrying of genetic information. How the double strand was able to explain the cell division, how the genetic information is transferred to two consecutive cells at the same time. The same genetic information is passed on to the new cells. And then we have talked about the amino acids, peptides and proteins, the basic structures, how chemical variations of amino acids, they dictate the secondary and the tertiary structure of the proteins. And now today we have seen how the protein folding and unfolding occurs. Finally, what are the structure of the fatty acids, the self-assembly and the lipid bilayer formations that forms a major constituent of our cell membrane. So, with this I will complete the module 1, and then the next module would be around, I will start some organic synthesis with the next module, then again after that module we will come back to biology. Thank you.